Good evening, my fellow peers. In our video, we will be talking about Fragile X syndrome and meiosis. Fragile X syndrome is a genetic condition which relates to meiosis, in which we will discuss first. Meiosis is a type of cell division that is present in sexually reproducing organisms that consists of 2 rounds of cell division but only 1 round of DNA replication. It results in cells with half the number of chromosome sets as the original cell. First stage of meiosis 1 is prophase 1, in which duplicated homologous chromosomes pair and exchange segments. Crossing over occurs in this stage. The next stage is metaphase 1, in which chromosomes line up by homologous pairs. The third stage is anaphase 1, shown here, in which each pair of homologous chromosomes separate from one another. The last stage is telophase 1 and cytokinesis, in which 2 haploid cells form, and each chromosome still consists of 2 sister chromatids. So the first stage of meiosis 2 is prophase 2, in which a spindle apparatus forms. Each chromosome will still have 2 chromatids associated at the centromere and move toward the metaphase 2 plate. The next stage of meiosis 2 is metaphase 2. Due to crossing over occurring in meiosis 1, the 2 sister chromatids of each chromosome are not genetically identical. Their kinetic cores are attached to microtubules extending from opposite poles. The third stage of meiosis 2 is anaphase 2, in which the proteins that hold the sister chromatids together at the centromere finally break down. This allows the chromatids to separate and move toward the opposite poles as individual chromosomes. The last stage of meiosis 2 is telophase 2 and cytokinesis. Nuclei form and chromosomes start to decondense. Cytokinesis occurs. This results in 4 daughter cells that each have a haploid set of chromosomes and are genetically distinct from one another in the parent cell. A meiosis recombination occurs. This is known as crossing over, which occurs in prophase 1. This is the genetic rearrangement between non-sister chromatids involving the exchange of corresponding segments of DNA molecules. This allows for genetic variability amongst the offspring. Fragile X syndrome is inherited in an X-linked dominant pattern. Over here we see a normal X chromosome. Over here we see a fragile X chromosome. As you can see, there is a mutated gene, which is the FMR1 gene. If you were to look at a karyotype and you're looking for the fragile X chromosome, you would find it at the sex cell, specifically at the X chromosome. As you can see, there is a gap over here. Here is the pedigree of Fragile X syndrome. As you can see, the black symbols represent those that are affected and those that are blue are unaffected. Symptoms of Fragile X syndrome, which include intellectual disabilities, attention deficit, hyperactivity, anxiety, unstable mood, autistic behaviors, sensory integration problems, speech delay, and seizures. Symptoms of Fragile X syndrome include behavior therapy, sedatives, muscle relaxants, antipsychotics, cognition-enhancing medications, and specialists. Such specialists include pediatricians, medical geneticists, speech therapists, occupational therapists, clinical psychologists, physical therapists, and neurologists. Everyone of every race and gender are affected. Males are more likely to be affected, in which 1 out of 4,000 males are affected by Fragile X syndrome, while females are affected from 1 out of 8,000.
For this part of the video, we're going to be talking about the cancer anaplastic large cell lymphoma. But before we can get to there, we have to talk about the growth of the cancer. We'll talk about mitosis and the cell cycle. Alright, so for the cell cycle, the first phase is called the G1 phase, where the cell increases in size, it prepares to replicate its DNA, and it grows. For the second phase, it is called the S phase, where the cell replicates its DNA, has 2 complete sets of chromosomes, and then for the G2 phase will be the last phase for the cell cycle, where the cell just continues to grow, and from there it will enter the mitosis phase. In mitosis, the first phase is prophase, where the chromatin fibers become more tightly coiled, condensing into discrete chromosomes. The second phase is prometaphase, where the nuclear envelope fragments and the microtubules extend from each centrosome and can now invade the nuclear area. Then comes in metaphase, where the chromosomes convene at the metaphase plate, a plane that is equidistant between the spindle's 2 poles. The chromosome's centromeres lies at the metaphase plate. And then comes in anaphase, where the 2 liberated daughter chromosomes begin moving towards the opposite end of the cell as their kinetic core microtubules shorten. And then comes in telophase and cytokinesis, where in telophase the 2 daughter cell nuclei form in the cell, and the nuclear envelopes arise from the fragments of the parent cell nuclear envelope and the other portions of the endomembrane system. And in cytokinesis is essentially the division of the cytoplasm that happens late in telophase, and it occurs from the formation of a cleavage furrow, which pinches the cell into 2. Alright, so growth factors. In order for the cell to divide during mitosis, it needs to receive a signal from a specific molecule, typically a growth factor. As seen from this picture, there's like a ligand and a receptor, and it will receive a signal in order to regulate the mitosis and the cell cycle. Anaplastic large cell lymphoma is a rare type of non-Hodgkin's lymphoma, with this cancer being caused by a mutation in the genes that produce white blood cells called T cells. However, this mutation is not always specific, as it can always be like a point mutation, meaning that there's a base pairing error for the T cells, therefore leading to them create a buildup of T cells in the lymph nodes, as shown here. Another thing to remember is that normal healthy cells also acquire two things: density-dependent inhibition and anchorage dependence. First, density-dependent inhibition is when crowded cells stop dividing, which is done with the cells sending signals to the surrounding cells once they have overcrowded to stop dividing, once they have formed a single layer and reached a certain density, as shown here from a little bit of couple cells to all couple cells. However, for cancer cells, they don't really follow density-dependent inhibition and they just keep growing abnormally. Alright, so for anchorage dependence, this is when a normal cell is being attached to a substratum, such as the extracellular matrix of tissue, to divide, which prevents the cell from dividing and floating off into the body once they have lost contact with the substrate to avoid piling off, as shown here. However, cancer on the other side doesn't really follow this and just creates a buildup of cells and creates this mass. This disease is transformed from a mutation in the genes that produces CD30 antigen, causing the cell cycle and the mitosis of CD30 antigen to not be regulated and abnormally produced, thus uncontrollably producing abnormal T cells, therefore creating a tumor in the lymph nodes, as shown here. So for the underlying causes, there really isn't an actual cause for this disease. However, we do know that it's not inherited and that the disease is provoked by a mutation in the genes that produces CD30 antigen for T cells. So benign is cancer cells that won't spread throughout the body or the tissues. However, malignant are cancer cells that spread to other tissues and organs, with this disease being malignant. Alright, treatments and prognosis. So the overall treatment of the patient depends on the stage of the lymphoma and the patient's general health. Some of the treatments they can use is chemotherapy, which is the use of anti-cancer cytotoxic drugs to destroy cancer cells. They can also use other treatment such as steroids, which are drugs that are often given with chemotherapy to treat lymphoma. They can help make chemotherapy more effective, and they can also help you feel better and can reduce feeling of sickness. They can also use radiotherapy, which uses high energy rays to destroy cancer cells while doing as little as harm as possible to your nearby healthy cells. And the prognosis of patients is about a 90% chance of surviving for 5 years with occasional spontaneous regression.